Tuesday the 11th, we're reviewing for Wednesday.
Yeah.
Okay, so we just finish class at 12. I mean, yeah, 12, right?
You mean on
On that Tuesday, because we don't have labs.
oh, well, we'll still go, yeah. Well, we go to 1 normally though
Yeah.
for class, so,
Yeah.
yeah. And then, yeah, you're, no lab on week. What happened to that side of the room today? Or the front? All your classmates are missing. All right, let's get started. So just a reminder, right, we have our poster presentations on Tuesday during lab. So that is everybody. And so it'll be fun. Like I said, we'll have, some refreshments. I'll bring in cookies or something, or fruits, things like that. And we'll do a poster presentation, so that should be lots of fun. And then if you need any last minute help on that, so tomorrow I have a lot of meetings, so I'm not sure I would be around. But Monday, you could always come to my Chem 104 lab section at 9 a.m. and get some last minute help then. All right, other announcements, questions? We kind of finish up. And of course, last wet lab this week, half of you already did that and checked out, so yeah, moving on with the quarter. All right, let's just dive into the rest of the lecture then, and then I have a fun demo, hopefully, if it works. And so our last little bit of this chapter is we want to focus on our carboxylic acids. So we got a quick introduction to them. Gosh, was it first term, right, when we mentioned acids and bases, and we kind of talked about the weak acids. And so this was the structure of a lot of our organic acids like we saw. So vinegar, right, we could draw vinegar back out. Just as a quick reminder. Oh yeah, this was vinegar. We talked about these acids. I can't spell anything today. And then, so we can get into a little bit of the naming. And so they take this ending of anoic, so oic acid. So if it's methanoic for one carbon, then it's methanoic acid. But just like before, we'll have common names. So this is usually referred to as formic acid. And then there's vinegar. So vinegar, the IUPAC name would be ethanoic acid. So remembering back to vinegar, it had two carbons, or of course acetic acid, vinegar, etc. So common name. And so the next piece of the puzzle to kind of wrap your heads around synthetically and chain it together is now we can take our aldehyde from before and oxidize it, and now we make our carboxylic acid. So this is how we do our synthesis. And so an example is just starting from formaldehyde or methanal, right? So starting from our aldehyde, so with our hydrogen there, again, hydrogen kind of being the key point that we can have something to leave, and then we can add our OH group on there. And then again, just kind of showing how it adds. So again, this hydrogen has to leave, so we have to have a hydrogen to leave. And ethanal, or acetaldehyde by its common name, but we can see ethanal, right, the aldehyde, and then it gets oxidized to our ethanoic acid or vinegar. And so then this is just a reminder of the synthesis. And so a recap from Tuesday. So we said we started with a primary alcohol, and that could undergo one oxidation to the aldehyde. And remember, the primary alcohol had those two hydrogens. Then it lost one. Okay, so now we make an aldehyde. Then another oxidation process to our carboxylic acid. So that's a common synthesis. And again, starting from the primary alcohol, two oxidations to give you your carboxylic acid. And so other things that we've touched upon in the past with our acid bases chapter is this whole resonance stabilization and why this is a good acid. So this is just, right, a recap of this. We can go through it so we can remember. Again, it's always good to practice our arrow pushing and organic chemistry mechanisms. And so we start off with our alcohol, and then, okay, we said, why is this the acidic proton? And then we can have our water acting as our base, just like we did in our acid-base chapter. And then action always starts from the electron. It's going to deprotonate, and then electrons go back to the oxygen, which then gives us one of our resonance forms here. And so now I'm just drawing this, so with a negative charge right on our oxygen. And then we said this was really favorable because it was resonance stabilized. And so again, we're just saying that the true nature is the average of these two structures. So this negative charge is distributed amongst those two oxygens. And then we can draw that with our arrow pushing and just show, okay, let's make a double bond. And then of course we can't have 5 bonds to carbon. That pushes those electrons up. And then we can draw our new resonance structure that just shows our movement of electrons. All right, so that's just reproving why that is a good acid, why this is the acidic proton, and it leaves. All right, so now let's look at our IUPAC nomenclature of our carboxylic acids formally. And so like we kind of saw, we just add the anoic acid at the end, or at least for our chains. So we have propanoic acid, so 1, 2, 3 carbons there. 2-chloropropanoic acid, and priority, of course, goes to the carbon on the carboxylic acid, so 1, 2, 3 there, so there's our 2-chloropropanoic acid. So same thing, 3-methylbutanoic then, so find our longest chain, 1, 2, 3, 4. So there's our bute part, 3, so methyl on the 3, so 3-methylbutanoic acid. And then just looking at this more complicated one, so right, not too tricky though, so 1, 2, 3 carbons, so there's our propanoic chain there, 2 chloros on there at the 2 position, and then another chloro on the 3, so that's where we get the numbering, right, 2, 2, 3, and then trichloro-propanoic acid. So pretty similar to the naming we've kind of been following. Questions on these examples? Seem okay. And so same thing, we can have trans versus cis distinction. There we have our trans, and then now we have to figure out how to label it. So again, priority at this carbon, 1, 2, 3, so there's the 3 for our ene, so pentenoic acid. So watch that there, right? So just the subtle E there for our double bond. And then looking, so now this methyl group, so this is cis here, and so now cis, now we can count it again, so 1, 2, 3, 4. So again, these methyls on the same side, and so now our double bond is on the 2, so 2-methyl 2-butenoic acid. And so right longest chain was still 4, have the E for the double bond, 2 for the double bond position, and of course, a 2 with the methyl. All right, questions about these so far? All right, so let's try some other stuff now. So now if we have rings, so let's draw a ring. 1, 2, 3, 4, 5. And so now the naming changes a little bit. And so now we add essentially carboxylic acid. So we'll practice naming. So if we just change our ending to to carboxylic acid, then this would be cyclopentane for our group here. And then now we just say carboxylic acid. And then stereochemistry could be an issue. So let me draw an example. And so I'm drawing it with a fat wedge there to show stereochemistry. So right, it's coming out towards us. And then we have our bromine also drawn with a wedge. So they're both coming towards us. And so they're on kind of the same side of the ring, right, kind of both flipped up. And so these then, since they're both wedged and up, they would be cis to each other. And so now we can name it. And of course, this is going to take the priority of 1 attached there. So now we just need to label the position of our bromo. So now it's going to be cis 2 bromo, and then cyclohexane. So of course, right, this is cyclohexane. So cyclohexane, and we add our carboxylic acid. Let's look at other aromatics. And so aromatics follow similarly, but they can also have special names like we've seen before. All right, take a minute to try to write probably the IUPAC name for it. So what might be the name of this one? What's this ring? Phenyl, or what's its other name? So just like the common one. So yeah, I know so phenyl's a group, but then what's just the ring? So benzene, yeah, so then it's just called benzene carboxylic acid for its IUPAC name. Or it has the other name of benzoic acid. So benzoic. So this is the common name. Let's do another one. See if you can guess this one. Any guesses for this one?
2-hydroxyl benzoic acid?
So 2-hydroxy. Did you say benzoic acid?
Yep.
And so there could be, right, permutations then, because you could also say 2-hydroxy benzene carboxylic acid. Or anybody come up with other permutations of how you might call it? Do you remember what this group is called? Yeah, a phenol. And so another name, and let me double check the spelling. So phenolic acid.
But can't carboxylic acid group be on different positions? Or is that just saying?
Yeah, so you'd probably want to specify like 2-phenolic acid, but you could also just, refer to this as the general groups of phenolic acids. And then did this look familiar to y'all by any chance?
Yeah, that's the salicylic acid, right?
Yep, so now it has another name too, and I can't spell that one either. And so, right, it has that common name, salicylic acid. Okay, let's try a few more naming. We'll practice another hydroxy one. If I can count this. 1, 2, 3, 4, 5. Do we have a guess for this one?
3-hydroxy pentanoic
3-hydroxy pentanoic
acid.
acid. That's good. So 1, 2, 3, 4, 5. I'll just write it. And then 3 for the hydroxy position, and then our pentanoic acid. So that looks good. No questions. All right, let's see if you remember that. Oh, sorry.
Because I thought it was 6 right there. But I might be counting off.
So 1, 2, 3, 4, 5. I'm not sure. Yeah, double count.
It might be the end for like the hydroxide.
There's no carbon next to the
Yeah, so
oxygen,
this
it's
is
just a
attached.
branch off to the
Okay.
oxygen.
Okay, makes sense.
Just like how for the hydroxy group, there's not a carbon up there, it's just branching off to the oxygen.
Okay.
But you're right, that's confusing.
Yeah. So those are.
Oops. So see if you remember the special name for that. Let's hear some guesses. So did we remember the name for this group when it's with other functional groups? If you don't, you can at least tell me the ending. What did you say? Oh, so what's the parent chain then? Of, yeah, this one. Like, what's the main acid? Yeah, well, you can still, so it's still our carboxylic acid, so we can do butanoic acid, right? For at least the ending. So 1, 2, 3, 4. But then what was this called? You remember? When it's not, it was that special name when it's like not the dominant functional group. And then I took it other, oxo, yeah. So yeah, 4-oxo-butanoic acid for that one. So it has that special oxo. So then oxo applies here. So what do we get for this name? So 2-oxopentanoic. So 1, 2, 3, 4, 5. Good. So pentanoic acid.
So you start counting off from the side which has hydroxide and oxygen?
Yeah, so the carboxylic group takes a priority in this case, and then, yeah, our ketone has to follow that. So that's also why we use the oxo, since the carboxylic acid gets the priority. So yeah, we always start the 1 for the carbon of the carboxylic acid. And then we can put other groups in there as well. So let's do some other little practice. So we have this group, which I won't spoil the name yet. So how do you name it? Then I'll draw another one while you finish. Any guesses for this top one here? I like the, so you had amino, but then count your carbons, because 1, 2, 3. Yeah, so that's just linked. So yeah, it should be 3 amino, and then what's the ending? Propanoic acid. And I'll give you another minute or so for that one. Guesses for this bottom one?
That one just be 2
2
amino butanoic acid.
amino-butanoic acid. Good. All right. Questions about those ones? Feel okay? And so let's look at a few other scenarios. So then what happens if you have 2 carboxylic acids? So let me see if I can draw this out. Give me a second. And so let's just examine what I drew here. So we have 1, 2, 3 carbons. And then, of course, now we have 2 carboxylic acid groups. And so now we can name this one, so propane. And then now dioic acid. So you add the di there. So then for comparison, so then this acid, we can follow what I drew. So 1, 2, 3, 4 carbons, 2 carboxylic acids, and so this would be named butane dioic acid. So just that di again. All right, let's do some fun naming. Do some harder ones. Okay. Try those 2 out. So what do we get for, let's do the one on the left. Wait, 2 hydrox? Oh. 3-hydroxy-4-methyl benzoic acid. Excellent. So that one probably wasn't as tricky maybe as this one, so you have to just make sure that our OH gets our next highest priority. So we have to label it this way, otherwise write 1, 2, 3, 4, and then 5, so that gives lower numbers.
Could I also call it 2-methyl-5-phenolic acid?
2, sorry, I was trying to follow you.
Because when you're doing phenolic acid, you count from the OH group. Right?
Oh, yeah.
So it'd be 2-methyl-5-phenolic acid, I think.
I feel like something's off with your counting. Because this, if you're calling this 5,
Calling
Ew.
that 1.
so this would have to be 4-methyl still. But
No, the OH group I put as 1. Should I put the carboxylic acid group as 1?
yeah,
Okay.
because then that would be 1, 2, 3. So yeah, because then you could do the 5-phenolic. But I think that's kind of confusing. This is probably better.
Okay.
So you're not trying to distinguish the relationship. And then what about this one? So I see in the chat, so somebody put 6-chloro-3-nitro. But the problem with that is you could get higher numbers if you go the other way, right? So you go 1, 2, 3, 4, 5. Then you get 2 and 5 as opposed to 3 and 6. Does that make sense to people? Because you kind of have two options. So this would be the correct one, where you're just trying to get the higher priority. Also, chlorine should probably take higher priority since it has a larger mass after a carboxylic acid. But then you're also kind of balancing, well, 2 and 5 overall is smaller anyways than our other permutation of 1, 2, 3, 4, 5, 6. So probably the 2, 5 is better. So then that will be 2-chloro, 5-nitro benzoic acid. Right, that was pretty good. And so other kind of reminders about carboxylic acids is that they can make charged species. So that's all that's showing here. But we've also kind of seen that with our acid-base chemistry, right? We do our acid-base neutralization reaction, and we make our conjugate-base salt. So that's kind of shown there. So we've seen that. And then, right, that can happen in a lot of other cases. So that's just showing that. And we also have naming. So then you add this O8 if it's a charge. So then now it'd be sodium methanoate or sodium formate. And not sure why I included this originally. So this is again showing benzoic acid. And then this is terphthalic acid. And so this is also used in like polymers. So to make plastics. We'll probably look at condensation reactions next week or right now. So we'll look at a different flavor of condensation reactions and these polymerization reactions. So I'll save that for kind of next week when we talk more about polymers. But let's at least look at this condensation reaction. And so this is to make an ester group. And so an ester is just basically a carboxylic acid now, but it doesn't have an OH. It has just some sort of other functional group attached to it instead. And so esters are pretty fun. They're pretty fun compounds to make. You'll probably make them in like an OChem lab because they're pretty easy to make. But let's talk about, well, what is an ester? Why are they fun? And so an ester is basically your fun smelling stuff. So they smell good. So people like esters. And so I showed some examples. And so there's our structure. And so this is a rum kind of smell of our ethyl formate or bananas. So this is a fun one that people like to make with our pentyl ethanoate. So that's fun. Oranges, so apples, pineapples. So they all have all the fruity smells. So all the esters are fruity smells. And then especially this banana one, and I always bring up the banana one, because if you ever make it in OChem and then you smell it, you're going to be like, that's the Laffy Taffy banana. Because it's that like really synthetic smelling banana smell. So that's also, again, where they get the flavors from. And so the general synthesis is that you take your alcohol, and so this is kind of what's attaching, and then it adds to your carboxylic acid. So again, this goes away, condensation reaction, so you lose water, and then we form our bond here. And let me pick some better colors. So right, these leave. So this makes water. So that's where the water comes from, that H from our alcohol, and then our OH from our carboxylic acid. And then the alcohol, this O is what attaches to our ester here, forming our new bond. And of course you can see obviously that's still there. So ester synthesis. And again, just kind of pay attention where the groups come from. This could be maybe an exam question of like what carboxylic acid and what alcohol make an ester, and you have to match up the groups, so where they come from. And so again, right, your ester group, your OR, has to come from your alcohol. So let's just kind of highlight that. So as long as you can just kind of recognize the pieces, that's what we need to take away from this. And there's also other fun reactions that we can do. And so we could take our carboxylic acids, and so this would now be like fats or oils, so long chain fatty acids, and then add them with some glycerol and some potassium hydroxide, some base, right? And then that makes our little soap molecule. And then we can, of course, do the opposite reaction, so then you could break that bond again with a hydrolysis. And so a hydrolysis just means that we're adding water now. So then that would be opposite of a condensation. So condensation is losing water, hydrolysis adding it back in. All right, let's do a fun little demo that kind of goes along with what we've been talking about. And so let's pull up what we're going to do. And so I want to do Tollin's test for you all. So Tollin's test. And so Tollin's test tests for the presence of an aldehyde. So it has to have that H to pop off. And so it's usually done with sugars to test for the presence of sugars that can reduce or not. And so let's kind of look, though, at the explanation and what we're looking for and kind of why. And so kind of the key that we should start with, so I'm giving away what's going to happen, is that it's going to react with glucose. But what's kind of funny is, right, you see glucose drawn in all these different forms. But I think this form kind of explains it the best of what's going on. And so kind of where the aldehyde is, because that's the first question, is, okay, you look at the structure of glucose, especially if you look at this structure, and it's not really clear, well, where's the aldehyde that's going to react? And so this is a better depiction of what is going on. And so basically we're saying in solution, this little group here is susceptible to resonance. And so it says like 2% basically is going to be in this actual aldehyde form. But that 2% is what is going to be active and reacting with our species here, again, assuming it goes well, and that will plate out our mirror test. So that's what's going to happen is that our silver itself will get reduced from our oxidation, and then we'll see the silver plating out from this reaction. And then I also brought along sucrose, and so if we get to it, we'll compare sucrose. Actually, I have sucrose. And so the sucrose will not react. And so sucrose is, of course, a disaccharide, and so that means it does not have, right, our little alpha OH here to react. So it has this bridge, doesn't have that, so it will not react with our tolerance test. All right, so let's see if I can do this. We'll see how it goes. So I haven't done this reaction before. And so first we have to kind of make our little testing thing. And so we're doing it on a small scale. And so some safety notes. So the reaction mixture afterwards is potentially explosive. So I have our beaker here to quench it. And we're also doing it on a small scale. I just thought I'd let you know if you ever wanted to replicate it. And then this is apparently how they do plate mirrors, or at least historically. But I think to kind of get, like for telescopes, you need like a really, really, really perfect mirror. So I think they have to kind of modify the procedures a little bit, or you have to clean it. And so that's the other thing. So I haven't tested this reaction before. I've only seen it done. And so I'm hoping the glassware is clean enough, so we'll see. So first step, we have silver nitrate going in, and it says about 5 mils. So hopefully that's 5 mils. And then we have our sodium hydroxide, so about a 10% solution, or 0.5 molarity. So there's good. So we're making our silver hydroxide. Just making sure. Oh. Okay, is that better? So now we're making just more silver hydroxide and making sure that's pushed all the way. And so now we're going to add, make sure I got my sugar solutions ready. So now I stole the stock bottle of ammonium hydroxide. So it's super concentrated, so we're going to see how it goes. And so it says to add it until our precipitate disappears. Oh, it smells like ammonia, like, you know what ammonia smells like. It's really strong. Now it's supposed to be counting. It smells really bad. Add a little bit make sure. Let's see if it works. It's hard to tell how dissolved it is. And the ammonia is bothering me a little bit, I will admit. All right, so let's try to add this glucose. This doesn't work, I may need more ammonia. Come on, glucose. All dissolving, which is a good sign. It's going to be anticlimactic. Come on. Go, go, go. Add some more. I'm just going to add a little bit more glucose. Okay, we'll let it sit for a little bit. So it might need to sit. Like I said, I haven't done this one personally. So let it sit for a minute. Can't tell if it's fading at all. It's definitely getting dark. Now I can't tell what's going on. What do we think?
Do it.
I just can't tell what's happening. I think it might be plating. It should be the silver plating. I think it's going. Yeah, it's happening. All right, you can start to see it happening. I can kind of bring it around. So do you see that it's kind of going silver on the outside? So it's got, you see it's slowly plating. So you see like the shiny silver. Yeah, so you can see the silver plating out. So like I said, I didn't clean it because I was nervous to clean it and make it worse. So that could be why it's not plating nicely. You can at least see it's silvery mirror. So ideally, you probably want to clean your glass surface with nitric acid first. You can see it is kind of plating or silver. Oh yeah, that's looking better. Oh yeah, so now we can kind of see it's on camera better. It is plating our sides. It's kind of cool.
So is that only aldehydes that will do that, or other metals?
So mainly aldehydes, but that being said, fructose would give you a positive result because it can do some weird tautomerization in solution. But yeah, for the most part, it should just be aldehyde. Oh, it's shining on my camera. That's kind of cool there. All right, I'm going to probably quench this now. So now I'm just going to rinse our flask out. And so this should be fine. It just has silver on it. So we could pass that around, or at least just kind of look at it. So now I've got more silver deposited. So that's how you make, again, your mirror surface. So if I clean it and use like a better mirror. And you can do this on like a larger scale. But like I said, I was a little nervous since I'd never personally done this reaction. And they were saying that it is explosive, like the mixture after.
Wait, are we gonna explode it?
No, that's why I poured it into the beaker of water to dilute it. Yeah, because you don't want to, I don't know the time scale, like I said, because I'm not too familiar with the reaction intimately, but you just don't want to let it like sit there as a mixture, because it can make silver explosive stuff. So then I put it in here to finish quenching it, and then it says to kill it with some acid, so I'll do that. So I'll put a little bit. Whoa. So now I just made a bunch of silver chloride that I'm going to have to filter off. And let's check the pH. Let's see, we're like still pretty basic. So it says to bring it to a pH of 2, but I might have to get some stronger acid. All right, I'll leave this for cleanup. And then, so other kind of fun things, so if we wanted to clean this mirror up later, we'd probably add nitric acid to it, and then of course I would dissolve it like we saw with the copper, but then that gives off NO gas, or sorry, NO2 gas like we did with the copper, remember the brown nasty gas, so I would have to do that in a fume hood. So. All right, I will work on cleaning up my mess. And so in the meantime, I'll hand out your quiz. Actually, I should probably hand out papers before I clean out my mess. And then if you need to take a break, take your break. I just want to get you started.
Do you have any more of the labs and homework for this week?
Oh, I do somewhere in my piles. Lots of piles. Here. There's the homework. I found the lab. Do you have lab today?
Yeah.
Okay, worst case, I'll give it to you there. Oh, I think I found it.